But first, what everyone wants to do is to model human physiology. And to do that, common research models has been used, sort of animals or flat culture, to use human cells. However, when modeling human physiologies, these models have some limitations, and they actually don't do a very good job in predicting the in vivo situation accurately. Therefore, there is an important task, and it is to develop better human models. To have a good model, there are some requirements that should be fulfilled, and one is that it should resemble the reality. So the model should resemble what you want to see in the reality. It should also be reproducible, not only within an experiment, but also from time to time. Secondly, it should be long-living, so you can do chronic exposure, for instance. And it should be abundant and very easy to obtain. So you need a robust protocol to develop your model. All of these characteristics have been shown to be possible when working with the 3D cell culture. So now I will show you some of the characteristics of a 3D cell culture model of a human one. The first one is that it's heterogeneous. So as in a tissue, where some cells will be close to a blood vein and others will be far away, those two cells are slightly different because of the different nutrient axes. So it's a 3D model where the cells in the core are slightly different than the cells of the perimeter. It should also have a decreased proliferation rate. A tissue doesn't proliferate as fast as a flat 2D culture, and so shouldn't your model. Secondly, it should have relevant genetic markers or secretion to resemble the reality. And it also should look like a cell or the model. Secondly, it should do a job of accurate prediction. So it should accurately predict a known characteristic. However, in 3D cell culture, there are some limitations and some issues, or some things that can be challenging. And first, I would like to show you a few different examples of different 3D cell culture techniques. This is not a finished list, but it's some of the popular things. And it includes lone binding plates and also gel embedding, and some of the more modern systems like organ on a chip or rotary bioreactors. I would like to define these to a static and a dynamic category. And just recall this as I go through the next part of the presentation. So when culturing cells in 3D, there are two parts that are very important. And the first one is this structure formation. The second one is the nutrient supply. So if we look at the structural formation of a 3D model, you will start with a sample. And this sample you can aggregate in various ways. For instance, you can use hydrogel or you can use different plate types. However, there is a problem with these systems. And it is that the nutrient supply is limited because there is no movement of the media. When you don't have any movement of the media, you have this situation where you create a diffusion depleted zone where the cells that are the cells will consume the nutrients that are close to them, and they will also build up the metabolic waste. So even though in this static situation, it's very convenient because the cell have peace and quiet to aggregate, but they will eventually lack some nutritional factors, and they will also have a buildup of metabolic waste that can affect the cells in a negative way. Therefore, the need for dynamic systems have emerged. And in a dynamic system, you will have movement of the media around the constructs. A common way to do this is that when you have your passive situation where the cells will consume the nutrients and build up the metabolic waste, it will also have poor access to the gas or oxygen. So the immediate result will be that you decrease longevity and size of your constructs. In an active diffusion situation, the media will move gently around your constructs, the nutrients will be better distributed, the metabolic waste will be removed from your cells, and they will also have a good gas access. So you have a possibility to grow larger constructs. You can not only grow them larger, you can also grow them for longer. But it's very complex when you want to move media around cells that has to build a structure. But you would still like to go from a passive to the active or dynamic state. A common way to do this is to introduce movement. It can be with a steered bioreactor or with the rotation. However, when you introduce movement, you also introduce some shear stress. And shear stress can become a problem when you are growing complex structures and they are not as well defined, or they are not tightly conjoined yet, so they can break apart. So it can affect cell conglomeration, but it can also affect the reproducibility of your constructs. Therefore, I would like to show you this picture of the CLINOSTAT principle for cell culture. So the principle is that you have a rotary bioreactor, like the one you see in the video. This will rotate and balance the constructs inside, so you balance the rotation to the size of your construct, and it will appear as they are standing still. Generating this type of, or using this type of principle for cell culture, the cells will have movement of the media, but they will have a very low effect of shear stress. We have used this CLINOSTAT principle in the CLINOSTAR system. So the CLINOSTAR system is a CO2 incubator, which is controlled by this tablet unit. Inside, you have 6 axles where you can place the bioreactors. These axles are individually controlled, so you can have 6 experiments at once. In the door, you see 6 black windows. They are cameras, so you can monitor your culture as it grows without having to disturb the environment. The bioreactor itself, the blue beads you see on the periphery side, they are the humidification for your cell culture. And on the inside, the middle, the see-through center, that's the cultivation chamber of 10 milliliters. And this is where you will grow your organoids or spheroids or the constructs. The way you use this, it's much like a petri dish. So you can access the entire culture chamber, and you can also have very large samples in this bioreactor. So it also possible to have biopsies within. The CLINOREACTOR is very convenient to use, and in the next slide I'll show you a video on how you can extract samples from the system. They are Hep2 cells. This is our founder Christoph who's using them. So you can, if I continue on. So here he's opening the bioreactor, and then he is transferring some of the constructs he has grown. So this is a regular Hep2 cell that has been grown for 24 days. And then he can close the bioreactor, and he has secured his samples. There are various different ways of working this system, and it's not exclusive to cell lines. I will show you a common overview of how you can utilize the CLINOSTAR. So like all cell culture work, you require a preparation phase or a sample. And this sample can be either 2D cells, or it can be primary cells from tissue, or you can have tissue pieces. And most commonly, you will need an aggregation phase. So you will need to select if you want to seed single cell suspension inside the bioreactor, or if you want to aggregate them or embed them in hydrogel before you seed. After you have selected your aggregation, you will equilibrate the CLINOREACTOR, and you can transfer your samples. Then it's followed by an incubation phase. And you can see in this case, I've made it 18 days, and this is what a common HepG2-C3A model needs before it's mature. During this maturation phase, that can vary depending on the nature of your samples, you should benchmark that towards the native tissue you want to model. But during the maturation phase, you change the media, like a regular cell culture. You need to do a speed adjustment, and you can also split or sample your culture along the way to follow the development. If you remember from previously, I showed you this characteristic of a 3D cell culture. So it should have decreased proliferation rate, relevant secretion of molecules, and relevant gene expression levels. The ultrastructure should also resemble the tissue you want to model, but most important, it should have an accurate prediction. I just want to show you what an accurate prediction means and what I think about or mean about it. So this data was made by Christoph and Steve, and what they have done is that they wanted to test how accurately a 3D cell culture model cultivated in CLINOSTAR could predict the toxicity of these 6 common use drugs. So they looked at the in vivo lethal concentration from literature, all the published studies they could find, and then they compared to normal LD50 values of the drug. So how important is it? If it has a perfect correlation of 1, then the model is perfect. So the first thing they did was to look at the 2D cell culture that was available. How well did 2D cell culture predict the toxicity of these drugs? And you can see that it has a prediction rate of 0.55. So it's almost random if this 2D cell culture will predict the accuracy of toxicity for these drugs. Of course, the pharma industry are very aware of this, and the golden standard is to use primary human hepatocytes to predict toxicity. When you use primary hepatocytes, it actually does a very good job in predicting the toxicity of the drugs compared to the in vivo data that is available. And you can see the correlation is 0.75%. What is interesting is that when you use a 3D model, and it doesn't necessarily have to be from primary cells, but it has to be mature, you actually get an equally good correlation. So it appears that a 3D model is equally good at primary hepatocytes, the golden standard, to predict toxicity. Now I will show you a few of the different application areas where the CLINOSTAR have been used. So all of these categories that you see here, we have published data and application notes, but today I will only focus on brain, lung, liver, and intestine. And the first I will show you is this liver carcinoma. So this is the HepG2 cells, and this is what they look like from starting, and they have been seeded as single cells and as they mature. So we have shown 29 days here, but you can already see that on day 3, they are already starting to conglomerate. And then during the 20 days, obviously you will change the media as they need to be fed, but you will also remove irregular constructs. And that counts for both constructs that are large or have conglomerated together, but also constructs that are very small. So eventually you will have a culture that's very uniform. And you can see they have a size of approximately 1 millimeter. The inside of such a spheroid will look like this when you stain it. So here you can see this is stained with Darby and for tubulin. And what's interesting about this is not really the structure of the spheroid, but it is that you see that the Darby is actually all over the spheroid. So this is a cross-section of the spheroid that has been stained for Darby. And if a common conception in 3D cell culture is that you have a necrotic core, but in reality this core doesn't look necrotic because the nuclei are intact. So we think that there is more of an inactive core that is important for structure of the cells. But you can see the perimeter of the spheroid will be much more proliferative than the core. But if you separate the periphery of the cells and you seed the core cells as a single cell suspension, they will form spheroids once again. So this liver model, that is one of the applications that you can use. And of course, you can use various cell types to develop your new model. What I will show you now is how you can use these cells to do chronic studies. So here we are looking at a graph that shows time to ATP level. And this is actually done inside the bioreactor over a period of 10 days, where the spheroids that are 3 weeks old, they have been hit with paracetamol or Tylenol multiple times. And then they have studied how is the cells reacting to this treatment. And of course, when you treat it with a high dose, that is the dark red line, you can see that they only need 2 treatments before they are completely dead. And if you have this dose, they will need 4 treatments before they are dying. But the large doses are actually not what's interesting in this graph. The first thing that you should consider is actually the 0.625 dose, because that is a normal therapeutic dose for humans. So you can see in this, if you have the spheroids, you can not only hit them multiple times, but you can also investigate what happens when I hit with a therapeutical dose of my drug. How will the cells react? And this is much different to 2D cell culture because they do not react to a dose that low, and you can't hit them multiple times and do sampling like you can in 3D cell culture. So this is a benefit of having a suspension culture, and how you can study chronic toxicity like you would do in a mouse. Another application here is lung organoid expansion. And this was a recent application note that we just published together with Professor Wilfingseder from Innsbruck University. And they have used these commercial available organoids from hub organoids. And for their downstream application, it was very important that they didn't have any gel tracks left of these organoids. So they needed to deassociate them and get rid of the gel tracks. And after deassociation, they would cultivate them in CLINOSTAR and try to expand them, which they did successfully. You can see here is a stain where it is stained for actin and the nuclei in blue, and red is the actin. So the benefit of using these commercial available organoids is that they are already representing a heterogeneous cell or cell types. So they are not, it's different cell types that are within. That's also evident here, where you can see how different the nuclei are stained. They are currently investigating if this cultivation step will change the different cell types. So this is for lung organoids, but another application area is these small cell lung cancer models. And in this case, they cultivated the small cell lung cancer, and they first characterized it. But now what I want to show you here is that they, after characterization, so they looked at protein content and ATP for viability. They also looked at the adenylate kinase release for apoptosis, and they also checked the glucose consumption. And finally, when they established the model, they have also treated the model with a common drug for this small cell lung cancer. And you can see that they have treated for 72 hours, 2 different concentrations. And here are some pictures on what it looks like and how they have established that the cells are actually affected by this treatment. So they have measured the surface area, and you can see that it's quite evident that the cells are affected on this treatment. The third application area I want to show you is how you can use stem cells to create embryonic bodies. And in this case, we have used AGGREWELL plate to create the EBs, but you can actually do it inside the bioreactor as well. And the EBs are the basis for many different organoids. So I will show you here in the next slide how you can create cerebral organoids in the CLINOSTAR system. So in this case, they have created the EBs in the AGGREWELLs, and after 24 hours, the cells was seeded in the bioreactor in induction media, where it started the neural differentiation. On day 8, they were transferred to, or the media was exchanged to expansion media, and then followed the maturation phase in maturation media. So here it's shown at day 12, but they are maturing the organoids until day 42, but we still need the data. So this is just to show you how many different applications can actually be applied within a system. It's really just the sky is the limit for this. Here are the different resources I've used. You can go in and look at the publications, or you can look at the application notes that are related to this presentation. And this is what I had to show you today. So if you have any questions, please fire away.
혹시 오늘 내용 중에서 궁금하신 점이나 질문사항 있으시면 마이크 하신 다음에 이제 질문 부탁드립니다.
저 질문이 있는데요. At first time use the orbital shaking method. What is the big difference compared to orbital shaking method?
So the CLINOSTAT principle compared to orbital shaking, the first difference is that in the CLINOSTAT, or the CLINOREACTOR, you will have the media chamber would be completely filled. So the cells will not have any contact with the surface. So you get rid of a lot of shear stress that is between the surface of the media and the air. So when you have the orbital shaker and it shakes around, there's actually not only shear stress introduced on the organoids from the plastic, but it's also introduced on the air-liquid interaction. So it is a more gentle approach to use the CLINOSTAT.
답변이 됐을까요?
네. And I am concerned about contamination when opening and closing the lid, because the lid is so tight. It looks so tight.
Yeah, I see the point. So right now we have seen a lot of different contamination, but it has mainly been from the media. But we have never seen any cross-contamination from reactor to reactor.
Okay,
Secondly,
I see.
we have also created a tool. So you can, of course, open your reactor in the left bench, and you can use the tool so you don't touch any of the critical parts of the bioreactor when you open and close.
Okay,
And this tool is supplied in each box.
Okay, I see.
네, 혹시 다른 질문 있으신 분 있으실까요?
Someone asking chatbot.
네, 다시 한번 말씀해 주실래요?
채팅창에 질문이 있는데.
아, 채팅창이요? Can you see this?
I've just got a question about that. It says, have there been any issues with organoids or 3D cultures merging together within the plates during culturing when using this orbital shaking method? So in my experience, both with using the orbital shaking method, but also using the CLINOSTAT principle, because right now you have one system, or you have one place where you have the entire of your culture. So of course there is a risk that the organoids will merge together. But if you have that happening in the orbital shaker, there's also a risk that you will have it in the CLINOSTAR. In the CLINOSTAR, there is a possibility to control the distribution of your organoids much more accurately than in the orbital shaker. So the risk is slightly reduced, but it's still present.
So it's possible the PBMC culture with this CLINOSTAR? This is the first question was PBMC culture, so like primary culture.
Yeah, primary cells. Yeah, so I have not been able to include it in this presentation, but we have just received the preliminary data of both people cultivating a glioma biopsy and also a liver model. So the primary liver cells are actually quite interesting, because right now the preliminary data suggests that they can culture them for longer. So they have a longer experimental window before the cells will start to die. Of course, the CLINOSTAR is not, it doesn't work magic, but it can prolong the experimental window of it. So instead of maybe 1 week, you will be able to cultivate 2 or 3 weeks and use your primary cells for longer.
두 번째 질문은 this system is the air liquid interface culture. Is it similar or?
So I think air liquid, this is not very different to air liquid interface. But if you, for instance, like Doris Wilfingseter, she is also supplementing her studies with an air liquid interface barrier model. So I think for 3D cell culture, it's important that you should have multiple ways of addressing your biological question. So maybe one 3D model is not enough, or you might need 2 or 3 different ones.
네, 혹시 답변이 부족하시면 채팅으로 남겨주시면 제가 통역해가지고 추가 또 질문해드리도록 하겠습니다. 네, 그러면 오늘 이상으로 저희 웨비나 마치겠고요. 저희 웨비나 참석해 주셔서 감사하고, 저희가 끝난 뒤에 저희가 설문지를 보내드릴 건데, 거기에 이제 정보를 보내주시면 추가 더 질문할 것이나 그 이메일 주소 남겨주시면 저희 관련 자료 다 보내드리도록 하겠습니다. Thank you, Julie, for this presentation.
Yeah, thank you for listening, everyone.
Thank you.
Thank you.
Thank you. 감사합니다.